The two-way ANOVA allows us to look at two different factors at a number of levels for each factor. And what we have here are some data from a really nice project by Ingrid, who was looking to make things better for mink. And she was looking at an environmental enrichment study in a 2x2 factorial experiment. Here we have two different tube types, one plastic, one wire, and a different number of tube numbers as the second factor. So there's either one plastic or one wire tube, or two plastic or two wire tubes as the environmental enrichment shell for them to hide in. And four of the many behaviours that were measured, and these are percentages of the observations, were the animals in the nest box. There were 2 animals per cage, animals that were active elsewhere in the cage, animals that were spending time in those enrichment tubes, or animals that were sleeping somewhere that wasn't in their nest. And we're going to use those 4 behaviours. You can see they're continuous by this ruler icon in Jamovi against these 2 factors. Because it's a two-way ANOVA, we can't use the most straightforward one we used last time. So here for ANOVA, we're going to go to the second one, ANOVA again, ANOVA, and that gives us this interface, which is really nice. So the results are here. These will update as we go. Unlike some programs, we're going to have to work out the ANOVA results and the p-values, and that'll be separate from looking for the means and standard deviations, etc., which we'll use using our descriptive statistics of later on. But just to do the ANOVA test, we're going to pick out one of the behaviours. We're going to just do them in the order that they appear. So nest, I'm going to put that as a dependent variable, so that's what we're measuring, and we're going to compare that for the tube type, plastic or wire, and the tube number here. And then there were 10 days observation, so but we're not including that as a fixed factor. And here is our ANOVA table. You can see that tube type as a factor was not significant in affecting the nesting behaviour. It doesn't say anything at this stage about how much nesting they were doing, but all we know is there weren't any differences between those treatments. And also tube number 1 or 2 was also not significant, and the interaction between those two things was also not significant. So that's our ANOVA. If we were to do something with a few more levels and we're interested in post hoc tests, that can be found here, but this is only where we've only got 2 levels per factor. Post hoc tests are unnecessary. But if we had 3 levels and there was a significant difference, we'd look at that and we could investigate our post hoc test there for another day. So we're not going to look at those now. We're just going to repeat this ANOVA for our other 3 behaviours. Now if I just changed nest for active like this, it just updates this single table, but we've lost our nest results. So what I suggest is that if we're going to do these in order, we put nest there. That's our first table. Now if I right click this and duplicate it, gives me the same table again, but this is a different analysis. So if I click on this one, I can now update that for active, and you can see I've got nest, I've got active, and if I can duplicate that one again, I can update that for the tube one, which is in the data example. And then if I duplicate that finally for my 4th and final analysis, I'm going to look at where they were sleeping. Now really we should look at those in detail as we went through, but let's have a look. So there's no significant differences for tube type, tube number, or the interaction for this nesting one. And there's no significant, remember 0.05 is our cutoff, so anything less than that is significant. Nothing here for the active behaviours. But when we look at the time spent in the tube, whether it's made of plastic or wire wasn't important, but how many tubes there were, if there was 1 tube or 2 tubes, is important. There's a significant p-value there, and we'll follow that up later when we look at the main data set. There was no interaction there, and there's no significant differences, associations either with the sleep else between any of the factors. So that's our main 4 ANOVAs. So that gives us a p-value that we're going to use. but we do want the main data set. In Jamovi, the best way to do that for the two-way ANOVA is to go back to your exploration. So we're going to create a table. So here, exploration, we're going to go for our descriptive statistics here. And here we can do all four at the same time. So I can put the nest in behavior, I can put the active behavior in, I can put the tube behavior in there, and I can put the sleep anywhere else that's not in the nest in there as well. And when it's split by, I'm going to put my factors. So I'm going to put in my tube type, plastic or wire. I'm going to put my 1 or 2 tube types, and you can see that we now have this quite extensive table with a whole range of descriptives. Actually, we're only interested for our presentation in the main table is for the mean values and the standard deviation values. So here under statistics, I can get rid of the ones that I don't want. So I don't want this one or this one, and I don't want the median in this case. I might want it another day, but not today. And I don't want the minimum or maximum. And you can see that simplifies the table marvelously. So here we've got mean and standard deviation, and you can see that it's very nice and clear, and you've got your contrasts. And that works really well. What you want is the variable across columns is quite a neat way of doing it. By default it looks across rows, and that's not so good. So make sure you've selected variables across columns, because that's just right to take into Excel for further analysis. So to get all of this analysis, so we want all four ANOVA tables and the descriptives. If I right click, I can copy all of it here. I could export it, but I'm just going to copy it. I'm just going to go to where my data came from in the first place, and I'll just post it here. So if I right click, you can either do it with all the details, or I like a nice clean version, so I just take the match destination formatting version here. So and there's all our values. Make the column a bit wider so I can see what we're doing. So for the table, we need these p-values. So we need these p-values here. We need these p-values here. Where it says 1, make sure it's 1.000. You need to have them all to the right and consistent decimal places. Here we've got the p-values, and remember there's the significant one there, and then we've got our values there too. Okay. So we don't need those references, so we can delete those. But here we've got our descriptive tables. We've got our means and we've got our standard deviation. And what we're going to do now is create the table with all these data. Okay. So what we can do, because actually what we want are our behaviors down the side and our tube types and tube numbers across the top. What I would tend to do is get everything sorted in Excel and then take that into Word, and I'm going to show you that in 2 stages. So let's have a look at it in Excel first of all. We've got all the numbers in a decent table. I mean, there's nothing wrong with that particularly, but it's not the preferred way of reporting. So here I'm going to copy everything here, copy, and I'm going to paste it. And I'm going to use this transpose function in the special. Did you see that? So when I right click, it gives me these 4 options, and there they are. And actually this way round, with the 4 behaviors down the side and the factors across the top, is my preferred way of working. And we've got some nice tips to work with that as well later on. Let's just make sure that they're all to 1 decimal place, because we've got our data to no decimal places, so our mean should be to 1 more. Sleep else look is only 22, so we want to make that 1 more decimal place. There we are. This one is, these are all at 2 decimal places for some reason, so we want to make those to 1 decimal place as well. There, I think they're all good. So and we've got the means and the standard deviations. We're actually going to combine those together, so we're going to put the standard deviations, we're going to put those underneath like this. And just for the actual table, I'm going to move this down here. You can see why I want this, because what I want is a row of means, and then I want the appropriate So these are the actual behaviours. I could put them there so they line up. I want these values to sit underneath the means, so the standard deviations and the means go together here. And then for active, let me create a new row underneath there. I'm going to take those standard deviations and put them underneath the means again, copy and paste, and here I'm going to move this down one, and I'm going to put those standard deviations there. I can put those, so I'm doing a Control C, Control V there, and that's getting it all ready for the analysis here. What else do I need? Well, I also need the p-values. So let's get the p-values in. So the p-values come from the table. So here I've got 3 sets of p-values. I've got one for the tube type. I just borrow that from there. I've got one for the tube number. I can put that there. Let's make those a little bit wider so you can see them. And then the third p-value is the tube type times the tube number for the interaction between those two. That's something that we'll have studied in class. Okay, so that's everything here. So what I've got up here for nest, I will need the 3 p-values. Here they are. I need to copy those. And at the moment they're in the wrong order, so I need to put the first one there, the second one there, and the third one there, which relates to the tube type 0.684, tube type 0.684, tube number 0.427, tube type 0.427, and the interaction one. Okay, so that's the first one, and I'll just take a pause and then fill the rest of them in. So after some frantic copying and pasting, here they are, and I've tidied it up a little bit. I've put it in green to show you what we actually need, which we don't need the mean word, and we don't need this other detail that we copied above. But here all I've done is centre it and just a bit more sensible spacing. But that's what we need to take into Word. So I'm going to take this and I'm going to copy this, right click copy, and we're going to take that into a blank Word document to create the table. But just to show you where we're going, I'm going to show you the table first. So here's the actual table. This is what it should look like when we're finished. Here we've got the table with its only horizontal lines, we've got the means, and we've got with the standard deviations in brackets, and we've got our p-values. There's just a number of things on here in formatting that I wanted to show you because I couldn't find this on the internet, so I thought it'd be worth seeing. So that's where we're going. Let's show you how we get there. So remember I've copied it to the clipboard. So I'm going to go to here, I'm going to right click. Now I could paste it in all green like that, but actually I'm going to go for using the destination style, which actually has been quite useful if you're going to do this in a thesis because it means all the fonts are already correct. So here, use the destination style. And you can see at the moment it's all looking a little bit confusing. So let's start with a bit of tidying up. Let's start with getting the behaviour terms right, and I know that some people forget to do this, and you can see everyone's tried really hard with their dissertations, but on their graphs or in the tables they haven't changed this from the original data analysis spreadsheet. So active nesting behaviour is fine, active behaviour in tube, I'm just going to make that in tube, that's fine, and here sleeping elsewhere. That can be explained in a title or somewhere else, but we're just going to leave it short. And then we're going to make that column as narrow as we can. I want to try and keep them on one line for reasons that you'll see. It's not essential, but it's quite tidy. Here we've got tube type and tube number. I don't think I need the word tube here, so I'm going to delete tube and I'm going to delete tube here. And that's everything we need. It's all gone onto the right lines. It's already looking at a bit tidier, but we've got lots and lots of squares, and you notice that I didn't have that on the previous version. So let's do a bit more tidying. First off, we're going to get rid of all these borders. Let's get rid of all of them. So I'm going to select the table in the top left hand corner, and here or here, it doesn't matter. I'm going to go here and I'm going to click no border. I've got no borders at all. But that's not how it looked. What I want is a border above the table. At the moment you can't see any of the grid lines. I turned them off so it look a bit tidier above. You see it's a bit tidier up there. Let's put a bit of space between these two. But actually, by default, you usually if you click on the table, look, I'm not on the table, I click on the table, I get two new blue menus here. In table layout, I want to view the grid lines. That's going to make life a lot easier as we can see the table. They wouldn't print, but you can see them to work with here. And I'm going to select the top line. I'm going to put in home, I'm going to put a border at the top of that and a border underneath that. So I've got one above and one below the titles, and I'm going to put another one at the bottom of the table. And that's the only lines you would normally have on a table. But when you've got a two-way analysis of variance, you also want to put something between here and here. And that's the trick that I've just found out recently. So here, what I'm going to do, I'm going to merge plastic. I'm going to right click and merge those cells because I want plastic to sit above these two. I want to get rid of the space there so it's all on one line. I'm going to merge those two as well. And again, I'm going to delete that space so it's all on one line. And I want all the p-values is covering that, and that. Even though I merge this, it's not going to make much difference, but you'll see why I'm going to do that. And I'm going to delete the other spaces that came from those empty cells. So we've just got the one line here. And this is what's really clever. I want to put a line below plastic that goes over 1 and 2 under wire and type number here. So what I'm going to first do, I'm going to collect all those, I'm going to center them. That's already looking a bit better. But if I select those three, when I go to borders and shading, I don't want to click bottom border. That just puts a line right across. If I go down to borders and shading, here, at the moment, by default, I'm applying it to the whole cell, but actually, if I choose paragraph from this list here, like that, at the moment it's going to put it across the top. I don't want it across the top, I want it across the bottom. So I'm going to click the bottom one on and turn the top one off. And when I press OK, you can see there's some gaps in it. I just think that looks really tidy. You know, just to make it clear what I've just done. If I go to table layout and turn the grid lines off again, can you see these? Now you could put blank spaces in there. You can mess with columns and widths and add another blank column in between and everything, but that's a bit of a faff. So we're not going to do that. I thought was a really nice solution. Let's put our grid lines back on just for working with. Okay, so that's all pretty much there. We've got a very boring job to do, in that when we've got the standard deviations, we need to differentiate them from the means, and so we need to put brackets around them. Brackets around that one, brackets around that one, brackets around that one. So for that row, and then let's put a bit of space between here. Hang on, put a space between the nest box, nesting and active behaviour, and then we've got to put the brackets around these as well. Another pause while I finish that off. And our time travelling is complete, and here we can see that we've got the brackets around all the standard deviations that we're going to use later on. Still a bit messy. Look, these columns are all a bit, these ones are quite wide and these ones are varying in width. Let's tidy that up. So here the p-values, they're less important actually than most of the data, so we're going to try and make these as narrow as possible. Let's numbers the longest word in there. We get that there. Make that column as narrow. Oh, that's too narrow. So we'll go to there. Make type a bit narrower. That's fine. So we've got all that here. Next up, we want to get all these columns the same width. So if I select all of it. I think if I start in the bottom left hand corner and then move up, see when I started in the top and move down. Oh, no, it did it okay that time. Here, when I go to table layout, there's a distribute columns, and I click that there, and that just evens them out. So they're now 4 even columns, but the numbers aren't very well aligned. So I want to sort that next. So here, although we've got brackets and we've got numbers of different lengths here, it's actually quite sensible to do everything lined up on the full stop, on the decimal point there. Okay, so in Word, we do that with tab markers. If you click in the column, you can see that all the columns in the ruler are lined up. You need to be able to view the ruler. If you can't see that, it should come up automatically. If you can't see that, you go view ruler. So we're going to go to the tab markers. I'm going to select all of the cells in the first column with the data in here. And if I wanted, the normal tab marker starts off looking like this. Oh, let's go through them. A left tab marker, but we don't want the left one. We don't want the middle one. We don't want the right one. We want the fourth one, which is our upside down T with a dot, and that's a decimal tab. And if I put that in the middle of this column, just there, they all line up on that decimal, which is very useful. And here, I always do that for the same for the second column. Boom, put that there, they all line up on the decimal. And the third column, and then here for the fourth column. Now these are all fairly consistent, 0 dot something, so these ones I'm just going to center. That's fine. So that's all our numbers taken care of. And that's the main thing I wanted to show you, and how the formatting is done. What else have we got to do? We've got to put a sensible title on it. I'm just going to copy and paste that. Look, table 1, it's the behaviour of the mink that takes into account everything in there, provided by either 1 or 2 hanging tubes of plastic or wire, and the means and standard deviations are the data that are presented. So that's fine. I'll take that table title and use it again. Probably don't want too much of a gap between the table and that there. There's only one thing to do left, and that's TTXTN. What does that actually mean? Well, we need to tell people. So I'm going to put in a footnote. I'm going to footnote 1. So if I put a 1 there, highlight the 1 and make that superscript, and then underneath I'm going to put that 1 superscript. And then I'll take that again. Let's put that. I need to make that superscript like that. Is the interaction term between tube type and tube number. And there, just to show you, if I take those grid lines off again, got a nice finish. The only other thing that I did is there was one significant result, and I put that in bold. Home, I just made that bold like that. That just makes it stand out. And that's how to present your two-way analysis of variance in a very, very professional APA standard, with all the details on there. You'll find a whole range of methods for presenting two-way ANOVAs online. Some are better than others, but here we've got everything that we want really. We've got the means and the deviations in a nice clear table, in the same table as our probability values, and quite often you'll find those are split because, as you've seen, your statistics packages splits them, and people tend to keep them separate on some of the papers that I've read. I think this is a good version. Thank you for watching.